I should tell you that before Adam decided to come to Griffith University to do his PhD, he interviewed all the possible supervisors around Australia by email, and that was the first time I'd ever been interviewed to see if I was good enough to be someone's PhD supervisor.
Had to check you out.
Well, there are lots of subsequent checkings out, weren't there? Anyway, the other thing I should say is that Adam's spoken about fish already, and he's a pretty dynamic, hard act to follow. Anyway, I'm going to tell you some stories from the early work and ongoing work we've been doing on fish ecology in the boom and bust system that we know and well so love. And you're going to see a few slides that you've seen before, but I don't think it hurts at this stage of such an important meeting to remind ourselves of why we think the Lake Eyre Basin is so important. And actually looking at where it sits in Australia, you could just say it's the beating heart of arid Australia. But it is enormous. It's as large as the Murray-Darling Basin, and it's one of the world's largest internally draining rivers. And Lake Eyre is the 5th largest terminal lake in the world. And as we know, it's largely unregulated. It's not completely wild, but it has low impacts, very few exotic species, and very high conservation values. In fact, on a world scale, if you think of all those factors and the fact that in Australia, our species are unique to Australia. They don't occur anywhere else in the world, and many of them in this basin are unique within the country. So it is very special. And I've been engaged in working on it with Stuart Bunn and other people through a project funded by the eWater CRC, looking at, essentially we started to look at the role of refugia, and that broadened out to considering the roles of all the aspects of the flow regime, the high flows, mid flows and low flows. And Stuart has shown you some of this information about the length of time between spikes of flow, very high flows. This was the one coinciding with the 1974 floods in Brisbane. And, he pointed out that the length of time between spikes of flow at the max in this flow record is 21 months. And that's a pretty critical figure for survival of fish and other totally water-dependent animals in the Cooper Creek. So moving on, I just wanted to remind us about this figure, which shows us from a statistical analysis on big rivers of the world that the Cooper, the Diamantina, and if we'd had it, probably the Georgina, are the most variable, hydrologically variable flow regimes in the world. And I've just put up there some of the stats that we were used to do this analysis. Sorry, I'll get it in a minute. Figures like annual and monthly flow, the monthly, same month but from year to year, the variability of the high discharges, the number of flow pulses, and the percentage of zero flow months. These are all more variable here than in any other part of the world. So what does that mean in terms of habitat and living there? I've put together a series of photos to make the story of how the system changes in terms of flow and habitat. And then I'll build on that, what happens to the fish. Now these, I cheated a bit because I couldn't get completely sequential pictures in time, so I've just put the story together. And you can see that, well, up there in the far top left, that we had a very large flood in March of 2000, and we were able to go out and work in Cooper Creek in the following year in our early studies on the fish assemblages and turtles and other biota. And so we were able to see the effect of that very large flood, and then a period of drying down to some sites with hardly any water left. This is one of my own photos taken when we were doing our work on waterholes and fish assemblages. So some sites go even below that and dry out completely, but then a little bit of flow will come through and you'll get a rising channel flow and various processes start, and then those flows can actually break out into increased channel flow, more complex patterns of flow on parts of the channel. Then they may break right out of those channels and into sheet flow, and eventually a large flood will generate the top left situation on that diagram. So what did we do? We were interested first of all in the actual assemblage of fish in these waterholes. How many species, what species were they, and how many were there? What was the relative abundance patterns in a series of different waterholes around the catchment? And our effort to document the actual diversity of the fish species was quite intensive. We used a whole range of methods that you can see there, And out of that, we got numbers of species and the kinds of species in each waterhole. And we also did a lot of measurement work. We sat there on the riverbanks. You can see the team of us working there. All the big nets were cleared. The fish went into a bucket. We took a sample, measured the lengths, measured their weights, identified them, and then put them back into the river so they were not harmed or there was no very low mortality from our efforts. So from that, we got a whole series of sets of data, and one of them was this being Cooper Creek, the list of species in Cooper Creek, and we know from what Adam's told us that there are, well, actually 12 species in Cooper Creek, and that's actually quite a high number for a desert river. Some of them are very common, some are special to the Lake Eyre Basin, and one of them is unique to Cooper Creek, that's the Cooper Creek tandan, quite a rare species. In the basin as a whole, I'm told by the people who are out there monitoring it now that there are 33 native species, 12 of them are endemic, that's a high figure. And across these systems, only 2 are alien, goldfish and the gambusia, and there are a few translocated species, ones that have been moved from another catchment into this one. So on the whole, it's a precious resource, it's quite diverse, and low impact so far from aliens. So I know this doesn't show up well because we can't turn the lights off. I'm not allowed to be in darkness. I have to be videoed. So what you can't see here is some of the figures, some of the bar graphs that I'm showing that indicate the number of species we found in a series of waterholes, those ones from which we sampled so carefully, in April and in September. That was a drying period in this study. And what I got from that was across these waterholes, up to 6 species were lost from individual waterholes totally. They just were not there the next time we sampled. Well, you could talk about sampling error, but we did put a lot of effort into the sampling. So what this is telling us is that waterholes can lose this part of their species complement totally, but they also change very drastically in abundance. This diagram, forget the little graph on the right, is just telling us that the way that individual waterholes lose their fish is it's not just a uniform loss of water. There's a whole range of factors that influence what waterholes will lose fish and which fish are actually lost. And this little model tells us that the position of the waterhole on the floodplain matters. If it's far out on the edge, it's likely to dry up quicker because it hasn't had a recent high flow. And then the connectivity of individual waterholes back to sources of flow in the main channel influences which ones will lose species. Bank erosion index was a very high driving factor, and then the morphology of the channel, the kinds of habitat, shelter, wetted perimeter and depth, and so on. We didn't get any measure of biological processes because they're very hard to measure. So this sort of information can be helpful if we do want to actually identify the most important refuge waterholes to sustain fish. And it's not simply a matter of size and depth. So moving on, we did get very good information on changes in abundance across that time period from April to September, and unfortunately these colours are drained out of the slide, but on the right you can see some very tall lines with broken bits. That indicates that we got some very, very high catches of fish and other places lower catches. And so through a number of analyses, well, the first fact analysis that came out of that was that 93% of all of the individuals that were present in those 15 waterholes in April, 93% of them were gone by our second sampling in September. Now that figure of 93% mortality is comparable with figures from other dryland systems and even other tropical systems that dry down in a dry season. Individual species responded in different ways, and we do have data on that. Obviously, when you're losing species and you're losing numbers of individuals, there has to be some compensatory processes that restore the assemblages to some waterholes. Perhaps some are never restored. And those processes are movement, which Adam has talked about, and reproduction. And so I didn't do it. So just to confirm the story that Adam told you, if you can listen any more to me, many of the widespread species just breed all the time, even in those waterholes with very little water in them. And that's a very good strategy if it's unpredictable as to when there would be a flow. But 11 of the 12 species that we recorded in these waterholes do make use of lateral channels and floodplains if there is a flood. And they go out onto the floodplains and they feed and they grow. And then very large numbers come back into some of the waterholes in a particular location. And some of those individual species move down the system into other waterholes, moving on the floodwaters. For instance, golden perch, we know, moves beyond the original source waterholes. There's only one species in the system that seems to need a rising flow in order to spawn. That's Hurtle's tandan. All of the others can just tick along like the turtles do in the dry season, but that tandan needs the rising flow. So, of course, we've heard about the importance of high flows, and in this study we were able to confirm the role of floodplain inundation in the boom of fish production by working out a relationship between the total numbers of certain species caught in the waterholes after that 2000 flood and a surrogate for the area of floodplain inundated. We didn't have aerial measurements, so we used two measures: total floodplain width around each waterhole and effective floodplain width. And that second one takes into account that there might be little hillocks and so on, so there's a reduced area where there's an elevational factor. And that just shows us what has been shown in many rivers all around the world, that some estimate of the amount of floodplain area inundated and the food resources generate creates this boom, this huge boom in fish numbers. So what we're seeing is analyses on Cooper Creek fishes that echo a vast amount of work done in other kinds of floodplain rivers. So that is reassuring that we're on the same track in terms of our results. So here's the picture of the boom and bust process just recreated. I've just mapped onto this the bust when the drying waterholes can achieve up to 93% mortality or 6 species lost, through to rising flows, distribution, breeding, and ultimately possibly a boom if we get a large flood. Well, I think that confirms in a very dramatic way the importance of flow variability and the low flows, the medium flows and the high flows that other people have talked about. So what I want to comment on now is what might happen if we do have irrigated agriculture in a system like this. And I want to introduce you to something you're very familiar with probably, but it's been called the irrigated agriculture syndrome. It's like a disease. It's a series of things that happen all together and they interact and create, well, very devastating conditions for river systems surrounded by or downstream from irrigated agriculture. And they are, of course, altered hydrology. How that happens depends on whether you have a dam in the stream or on the landscape. Floodplain loss by not releasing large flows or not enabling large flows, alterations to connectivity, all those issues that Arthur talked about, slight changes in elevation that change the way in which water moves, connectivity and so on. Contaminants and nutrients are part of this syndrome, often riparian loss and degradation, and often alien species. We know that some alien species do very well in regulated systems. I've listed a few other threats there that I'm not going to talk about in detail, but obviously, possibly, fishing is one of them, mining, the elephant, and climate change, which Jenny will talk about. So what we need to be able to do is to understand some of the implications of those stresses associated with irrigation on measures of ecological health. So on the vertical axis, I've just put very unhealthy to healthy at the top, and I'm suggesting fish indicators. We'd have to change the indicator according to the issue. And the curves that I'm showing are just my suggestions of the kinds of changes that you might see in the health metric as you increase a pressure along that horizontal gradient. So the first one I've got there is a threshold relationship, this one here, which I think would represent either declining depth and change in character as you reduce the low flows in a system, the sort of thing that Stuart was talking about, or it might also represent a threshold in the change in number of waterholes that are able to act as refugia. So I think there might be a possibility of some change that's acceptable, doesn't alter the health metric, but then a probable threshold and a rapid decline. I think this linear one represents declining fish production off floodplains. If we made some changes on the floodplain, that's just echoing and extending the natural relationship that we've demonstrated. And this one I suggest here might represent just a fairly moderate level of some very toxic substance, a pesticide, for instance, coming off an agricultural system. And you might expect numbers of individuals of certain species to decline very rapidly in that circumstance. So these are just hypothetical relationships. Some of them we can probably put numbers on and use to predict the effects of irrigated agriculture. So without any further ado, I just want to mention that Harry Balcombe and I, who've done most of this work together, put together a synthesis of all of our understandings of the boom and bust ecology of fish in Cooper Creek. And at the end of it, I wrote many things, but here's a couple of things that I think are important. Preservation of natural flow intermittency, these patterns of low and high flows, sequential flood pulses, complex habitat mosaics, connectivity, and the actual environmental flow requirements of valued species are key scientific principles for arid zone rivers. And the final point, I think that these boom and bust floodplain systems are telling us a great deal about biodiversity protection and water management principles that could help us as more systems become drier and more stressed for purely aquatic animals, fish and turtles and so on. I had another point, but I can see Angus standing here is ready to drag me away, so I'm going to close it there. Thank you.